Okay, thank you. Welcome everybody to this webinar. We'll be giving an introduction to polygenic scores in 4 national cohort studies. We're joined by my colleague Tim, who has led this work, and by my colleague Liam, who will be talking about research examples. Just to mention, there's an asterisk here. This is because we'll be referring to polygenic scores. We also use the terminology of polygenic indexes or polygenic indices to refer to the same thing. First, housekeeping. Firstly, your cameras will be switched off and mics muted. If you have a question, please use the chat function. And please note that your question will be visible to all attendees. If you have any technical issues, please email us on this email address here. And finally, we'll be grateful for your feedback. We have a series of webinars and training materials, so please follow the link in the chat at the end of the event for the short survey on feedback. Thank you for joining us today. So the outline, I'll first give an introduction to the CLS cohorts for those that are less familiar, and talk about the value to research by incorporating genetics with cohort studies. Tim will then be giving an overview of how the polygenic indices were generated, and how they've been made available, which ones have been made available at CLS. We'll then give an overview of how you can access these resources, and then Liam will give some examples of research using these polygenic indices. And then we'll have the Q&A. So for those of you who are not familiar already, what are the CLS cohort studies? So they are birth cohort studies. So the most recently born was the Millennium cohort study. And around the millennium, around 19,000 individuals that were born around the millennium were included in this longitudinal study. They gave data shortly after birth. And then there's been repeated data collections throughout their infancy and childhood. And we expect that to continue long into the future. So each of these birth cohort studies has a similar design where data is periodically collected on individuals born in different birth years. So we have individuals in the Millennium cohort study born around the millennium. We have the cohort study, the Next Steps cohort, which was born around 1989. We have the 1970 birth cohort study, the 1958 cohort study, and then managed separately from CLS is the 1946 cohort study. And we'll be discussing these 4 cohorts in this webinar. So you can see on this slide, we have data across many different generations, and we have data across many different life periods on many thousands of variables, including health data, social data, demographic data, biomedical data, and so on. Our website contains a lot of very useful resources to give an introduction to the different cohort studies and the kinds of data that have been collected, like this URL here. We have webinars to introduce the cohorts. We have video series introducing more specific aspects of the data, for example, the mental health and well-being data. We also have methodological videos as well. So this one on how to handle missing data in our cohort studies, both with theory and demos. So please do take a look at this. So I think some of you here will be already quite familiar with the cohort studies, but you'll be less familiar with using genetically informed designs. And by that, I mean using genetics in your research. And so what is the value of genetically informed designs at a higher level? So at a high level, they can be useful whether you're interested is actually in the environment, or whether your interest is actually in the genes or some combination of the two. So one prominent example is the study of gene-environment interactions. You may anticipate that the influence of genes might differ by factors like age, the cohort in which you're born into, or other more specific aspects of your environment. Often in cohort research, we are attempting to adjust for confounding variables when we're analyzing associations. And we are limited to the variables which we have measured. And we're there often concerned about issues like unobserved confounders or residual confounding bias. So the use of polygenic indices gives us additional opportunity to account for confounding. There's a very active literature on adjusting for genetic confounding, which has thrown up some quite interesting and surprising results. So you can now do that using these data. You can also use genetics as an instrumental variable approach to try and get a better handle on causal effects. So you use the genes as an instrument. So for example, in Mendelian randomization analyses, to try and understand whether the effect of an environmental variable is thought to be causal on the outcome. We can also use genes to help with measurement. we can help use them to help approximate unmeasured exposures in our data, or exposures which are not measured ideally or measured inconsistently across different cohorts. Because genes have been measured and can be quantified in the same way, they can be helpful simply for getting a better handle on the measurement of the exposures that we care about. And a final point is that for those of you who are interested in building prediction models, either in the health sciences or the social sciences, genetics can be helpful when you're attempting to predict individual life outcomes or attempting to build models of great explanatory power. So on this slide, we have a series of papers giving illustrations of how genetics can be incorporated into sociology, into genetics, and over on the right here, in observational research more broadly, perhaps particularly here in epidemiology and psychology research. Also shown is a recent book showing how genes may matter for the study of social mobility. So we have here data which is very useful scientifically, but also very interesting substantively in terms of social inequality as well. So many of you in the webinar may actually be familiar with genetics, and you've been using the predominant genetic data resources, the biobank studies in your research. So the biobanks are extremely large studies, so they typically have sample sizes of hundreds of thousands of individuals, and they have genetic data. So in the context that you already have biobanks, why should you care and why should you use cohort studies? Well, the first point to mention is that we have very rich phenotypic data. This is a simplified timeline showing us the kind of range of detailed physical and mental health data that we have across the entire lifespan from birth onwards. We also have very detailed measures of social position, the grandparents, the parents, and the own individuals themselves. Similarly, we have detailed information on demographic variables, social variables, partnership variables, and so on. They have extremely rich data. The second point is that we have data across the life course, enabling us to study factors in childhood and early life that are simply not possible to study in biobanks established in midlife. And third, we can compare across different generations. So across over a 50-year period, we can examine how associations may differ. It may be the case that an association with a gene may in fact depend somewhat on the context in which that gene is expressed. We'll be hearing examples of that later on in the presentation. Number 4 is that these studies are designed to be nationally representative. So this can help us to mitigate concerns of selection biases that the volunteer-run studies are particularly susceptible to. So we have nationally representative sampling frames, but like all longitudinal studies, we have attrition that occurs across time, which can reduce statistical power and may bias your analyses. But thankfully there, because of the birth cohort design, we have the early life data to help us to use principled approaches to handle missing data. For example, multiple imputation. We provide videos, we provide code on our websites to help users do that. Number 5 is that we have in the Millennium Cohort study not only data from genetic data on the cohort members, the offspring, we also have genetic data on the mother and the fathers, giving us this trio data set of genetics. This could be extremely interesting. For example, you could examine whether it is the genes of the participants which are influencing the outcome, or whether it is that in fact the genes of the parents that are affecting the offspring's outcome, either transmitted genetically or transmitted via the environments in which the parents are cultivating. So using a within family kind of analyses as well as possible here. So we've given an overview of the scientific utility of the data in our recent data resource profile, which I'd encourage you to look at. It's been published in IJE earlier this year. And we are very excited about this data. In the past, our genetic data has been made available freely for research use, but it requires very deep technical expertise. The genetic data are extremely large, typically the size of an average computer hard drive. Whereas now with these polygenic scores, we're hoping to unlock a new era of genetically informed research in the cohorts. I'll now hand over to Tim to discuss how these polygenic indices were generated. Tim.
Okay, hey everyone. So I'm going to talk about the generation of the polygenic indexes now in the CLS cohorts. So first of all, I'm going to talk about genetic data. The Each of our cohorts have sampled our participants using either blood or saliva samples, and then these samples have been genotyped by the Bristol Biolabs. The data have been returned to CLS, and then we've run a series of quality control processes over the data sets. This is fairly standard QC processes. If you're interested in these, we go over these in more detail in the genetics webinar from last year, which you can access the QR code and the link on the right. And there's more detail on the GitHub that I'll come to shortly. After QC, we impute the genetic data to the top bed imputation panel. And what this is essentially the genotyping doesn't cover every single marker in the DNA. It covers a select set of markers, and the imputation kind of fills in the gap, if you will. Then after the imputation, we do another round of QC across the cohorts, and this is done in exactly the same manner in each of our cohorts, and also the 1946 cohort that was mentioned. So we end up with two kind of final sets of data set. The first is QC'd imputed data on each of our individual cohorts, and then the second is what we call a harmonized set, where we further restricted the genotype data to be the exact same across each study. The goal of that being that kind of cross-cohort work is a little more comparable. So that's the first step. Then if we think about who's in these data, so we've got a big table here showing across the CLS cohorts the ages that the samples were taken, total cohort number, the number who responded to the biosampling wave, etc. And this column here gives the total number we have imputed QC data for in each of the cohorts. And then we've calculated the response rate here, which is based off of the number imputed and QC'd of those who were alive and in the UK at the biosampling wave. Those are the ones who were eligible to participate. And then over on the right, we've got the numbers who we have PGIs available for. You'll note the little asterisk by the Millennium cohort, and that's because in the Millennium cohort, as David said, we've genotyped not just the cohort members, the offspring, but also the mothers and the fathers. This results in nearly 8,000 cohort member mothers, 4,500 cohort member fathers, and then from these three, we have nearly just over 10,000 offspring parent duos and just over 3,000 offspring mother-father trios, which offer really nice potential for analysis, which Liam's going to talk about later. Okay, little brief bit on polygenic indexes. We have on the right here, we have a hypothetical rung of DNA for 3 different people. You can see that most of the points of our DNA, the human genome, is the same between all people. At very few points, we have some differences. So I'm just going to move this box out of the way here. So here we see, for instance, a purple person has an A allele, a blue person has a G allele, and orange person has a T allele. And it's these points of variation that we're really interested in when we're kind of looking at human genetics. We can see, or we can use GWAS studies to try and identify how these different alleles, these points of variation, relate to traits of interest. So a GWAS, excuse me, a GWAS here will look at these individual points of variation, and it will say, how does, across the population, how does having an A allele associate with our phenotype of interest, say height? Now, each of these individual markers typically has a minuscule association in terms of its effect size. So certainly for complex traits, we see that's the case. But when we combine these effect sizes, they can kind of together explain quite a bit of variation, and this is what a polygenic index is. So here we've got a hypothetical run of DNA at 5 sites. So we can see our GWAS, this here is our GWAS estimates, and it says that if you have a C allele at the first site, that is associated with a negative 0.02 change in the outcome. If If you have a G allele at the second site, it associates with a positive 0.01 difference in the outcome, A allele at the third site, 0.02, so on and so forth. And now this hypothetical person, they have one count of the C, the effect allele, as we call it, at this first site. They have two counts of G at the second, one count of the effect allele A at the third site. So while all of these kind of individual SNP effects are tiny, we can summarize these effects. So at the first site, they have one copy multiplied by negative 0.02, the kind of effect estimate. The second site, they have two copies of G, so 2 multiplied by this effect size of 0.01. And we sum up all these numbers. And for this hypothetical section of DNA, we get a polygenic index with a value of 0.052. Now, we do this across an entire sample, entire population, and because the polygenic index is essentially kind of a sum of a huge number of accounts, it ends up being normally distributed in the population, and we say it's just polygenic index is just simply, or calculated this way, it's just simply a kind of a result of the count of effect alleles multiplied by their effect sizes from a GWAS. One important thing to bear in mind is that this is dependent on the trait and the population who are measured in the GWAS study. I'll come to this a bit later. So in terms of how we've calculated the polygenic indexes in this first version of the PGI repository, we've used a program which many of you might have heard or used yourselves called Precise2. And what this does is it uses what we call a clumping and thresholding approach to create the polygenic indexes. Excuse me. So in terms of clumping, where to demonstrate here, we've got what's called a Manhattan plot from a very large GWAS by Okpe et al. In 2022. This is a nice demonstration because we've got, there's a lot going on in the figure. So what we've got here, we've got these numbers here, 1, 2, 3 through to 22, represent the autosomes, so the non-sex chromosomes in DNA. And then each tiny dot on this figure is the p-value for an individual SNP and how it associates with the outcome of interest. In this case, this is educational attainment. And what you'll see is that there's these kind of really big spikes, hence the name Manhattan plot. So there's a lot of spikes here, for instance, a lot of, if we use the terminology, highly significant SNP effects kind of on the same point of the genome. Now, part of this is because these aren't necessarily independent SNP effects or independent SNPs that are related to the outcome. So when we inherit our genomes from our parents, we tend to inherit closer bits of the genome in clumps, if you like. So what this means is that these aren't necessarily all individual SNP effects. They could all be tagging the same causal SNP, whichever one it is in the genome. So to get around this issue, we clump the SNPs. So we do this using this fairly standard parameter. So we take the SNP with the largest effect size in a 250,000 base window, bases being the individual points of the genome. And then once we've done that, we calculate the SNP effects at each point of variation for all of our samples. And then the threshold part of this comes into play. We generate polygenic scores at multiple p-value thresholds. So this dotted black line here represents the what's often termed as GWAS significance, which is a p-value of 5 × 10⁻⁸. We use this highly restrictive p-value because it's essentially a Bonferroni correction, accounting for the millions of tests that we're doing when we do a GWAS. But what we might want to do is we might want to use multiple SNP thresholds for different reasons. So in this version of the repository, we've created polygenic scores, so polygenic indexes at 5 different thresholds. So we've got here what's often termed as GWAS significance, and then we kind of go all the way up to this threshold or a p-value of 1, so we include every single SNP. There's different reasons for using different polygenic indexes. this is kind of my opinion on it. Generally speaking, as you include more SNPs in a polygenic index from a GWAS, you tend to get more predictive power, including more information. Some of those signals might be a bit spurious, or they might be entirely spurious, but prediction doesn't care for bias. So if your goal is purely how well does this predict a trait, you might be more inclined to use more liberal thresholds. The stricter threshold polygenic indexes, you can consider them maybe as being more reliable. They're more likely to capture individual SNPs that are more reliably capturing a true causal effect on the phenotype. That's not absolutely always the case, but I think generally it's a good rule. So in addition to these 5 polygenic indexes, we've calculated, so this is for every trait, we've calculated at these 5 thresholds using the cohort independent data. So what I was explaining about the QC'd and imputed data set, for instance, for MCS, but we've also generated them on the harmonized cohort data sets across all the cohorts. Now, a really important point that we have to mention is that genetic studies, I mean, more so than most research studies, are heavily dominated by these weird, what's termed weird samples. So Western, educated, industrialized, democratic, and I've completely forgotten the R. So someone might be able to correct me in the chat. And what this means is that a lot of the samples that are used for genetic research, they come from overwhelmingly from, say, the UK, the US, and Scandinavia. There are studies everywhere around the world, but they tend to be smaller. And the result of this is that our genetic studies are overwhelmingly dominated by what are often termed as European ancestry individuals. So this is a lovely graph from a paper. It's a little outdated, but sadly the trend is still pretty much the same. And what we have here is we have these circles represent sample size. No, they do. I think they do. And the colors here map to this legend here. So you can see this kind of overwhelming array of blue dots, which corresponds to European individuals or European ancestry individuals, and these two kind of histograms here demonstrate the dominance of these people in these studies. Now, genetic ancestry is a incredibly complex and often misinterpreted term. We could get really bogged down in this here, so we're not going to discuss it in huge detail, but there's, in the slides we'll make available, there's a reference to a wonderful paper by Graham Coop, which explains kind of the difficulty of labels of European ancestry really nicely. But the take home for the current version of the polygenic index repository is that we've only derived the polygenic indexes for individuals in the CLS cohorts who are genetically similar to European ancestries in this really big, kind of well-characterized reference data set called 1000 Genomes Phase 3. So you need to bear that in mind when you use these, that the scores are only available for individuals who are often termed European ancestries that we can say are similar to those of European ancestry. Those of you who work a bit more with generating polygenic indexes might be interested to know, so we've got the full code pipeline to generate the polygenic indexes from the initial cohort genotypes all the way through to every single PGI. We have the code available on GitHub at this link. Everyone's obviously welcome to look at it and see what we've done. The whole point of this was to ensure transparency in what we've done. And those of you working in other cohorts, you're able to clone the pipeline, and it should hopefully be quite straightforward in terms of replicating it on your own cohort genotype data. Okay, so on to the actual PGIs that are available. We've derived 44 polygenic indexes over these 7 we've imposed domains. We've got the full list on the right on the table, and essentially these fit over these domains on the left. So for instance, in terms of anthropometrics, we've got things like BMI, height, for brain structure and cognition, have Alzheimer's disease, cognition, health behaviors, alcohol use and cigarettes, and so on and so forth. And on the right here, we've got a screen grab of our genomics data GitHub, and the QR code there will take you to this site. On the GitHub, which has information about all of the omics data in our cohorts, if you go to the Polygenic Indexes tab, you'll see, first of all, the links to access these PGIs on the UKDS, and you'll see each of the PGIs we have and the GWAS that it's been derived from. It also links to the code and the documentation that we've got for the derived PGIs. So in terms of PGIs to return to, so this, for each trait in each cohort, we have 10 PGIs. We have those just in the cohort dataset and those in the harmonized dataset. This table here shows that when you use the harmonized dataset, you tend to have slightly fewer variants going into the PGI. This number's not a massive difference, and we've done some testing to look at the impact this has on excuse me, impact this has on genotype phenotype associations, and the result is minimal. You see effectively the same effect size and variance explained regardless of which dataset you use. Here's an example PGI for height in the NCDS, so you can see it's normally distributed. This is the all-SNP PGI. And on the right here, we've got a table of results. This shows the variance explained in this measure of height at 16 years in the NCDS using only sex and the first 20 principal components of genetic variation. And this second column shows the explained variance when we add in the PGI. So we end up with this incremental R squared showing the additional variance that the polygenic score explains. And we see that as we go for, generally as we use more liberal thresholds in our PGI, we explain a bit more variance. In terms of what they look like, they're all the PGIs are available via the UKDS in a CSV spreadsheet. And this is a status screen grab, apologies to those of you who are users, which shows the naming convention of the PGIs. We've done this to try and ensure that there is as straightforward to use as possible, but also that in the event of queries from data users, we can very easily identify the variable you're using. And essentially the naming convention consists of the cohort, whether or not it's from the harmonized data set, the trait, the p-value threshold, and the repository version, so in this case, v1. Okay, quickly on to access and resources. There are 2 routes for access for the PGIs. If you want to use the raw genome-wide genetic data, or you're already using these data, you've got an application approved by CLS, and you want to add the PGIs in, you'll need to access the data directly from CLS through the this link. It consists of an application form and filling out some variable spreadsheets. If, however, you don't care about accessing the raw genome-wide genetic data and you just want to use the PGIs alongside the other data in the cohorts, you can access these from the UKDS. So this screen grab on the right shows the NCDS polygenic indexes. If you've not created an account, it's fairly quick to set up for the UKDS, and you can access the data. You have to adhere to some access kind of rules, and then there'll be a short application form to fill out to get the data released. And then you can combine these with the other data that exists on the UKDS. For those of you who aren't familiar with our data, there's a couple of different ways that you can search the general cohort data we have. The first of these is the closer discovery search, which you can find here. And this contains our data alongside other studies such as Ulsbach and Generation Scotland. And then the other way you can look at our data is if you go to the CLS data access page, you can see on this page we have variable spreadsheets for each of the cohorts. Different people have different preferences on how they want to look at the data. I personally quite like these huge spreadsheets because it allows you to see everything that exists. And then just for me to finish, here's a couple of resources. So the GitHub that I was talking about contains hopefully all of the information you could want about our omics data. We're treating it as a living documentation. And then this QR code will take you to the cohort profile that David mentioned, which explains the genomic data we have in our cohorts in the 1946 study. Okay, I'll pass over to Liam.
Thank you. So yeah, thanks, Tim. I'm going to talk about 3 papers where we've used polygenic indexes in the cohorts already. Chosen these because they exploit some of the nice attributes of the cohorts that David spoke about at the beginning. Sorry, it's not going on. Here we go. Okay. So the first study is a gene environment interaction study, which was led by Tim, and this looked at the association between a polygenic score for education and phenotypic achieved years of education, which used data from CLS's 2 oldest cohorts, born in 1958 and 1970 respectively, as well as the NSHD, which is a cohort born in 1946. Now, this period that the cohorts cover overlaps with a quite significant expansion in the years of education achieved in the UK by young people. So just looking at the result on the left-hand side, this is the association between the polygenic score and achieved education. The green line is the 46, blue 58, and orange 1970. And you can see that in each successive cohort at a given polygenic score level, there was more achieved education in the more recent cohorts, but actually the association between the polygenic score and the achieved education was stronger in the most recent cohort. We were also able to look at, because we've been able to create so many polygenic scores for different phenotypes, we're also able to look at a polygenic score for cognition specifically. And we didn't see such a change in association there, but it should be noted that this polygenic score is less highly powered than the one for educational attainment. Another nice characteristic of the cohorts is that they have lots of harmonizable measures. So you can pool cohorts together to run analyses, G by E interaction analyses that achieve greater statistical power. So here we look at the interaction between socio-economic class of origin and the polygenic score for education. So we've got 2 different measures of socio-economic background. We've got parents' educational attainment on the right-hand side and parents' socio-economic class on the right-hand side. And in each case, we see stronger, at a given polygenic score level, we see more achieved education, the more advantaged social background that you have, but we also see a stronger association between the polygenic score and achieved education. These data can also be used to examine potential limitations that are in standard observational analyses. So a basic finding of social stratification research is that social origins are in some sense sticky. People aren't completely socially mobile. Now, this could reflect a role of the environment, but it could also be reflective of the fact that we inherit genes from our parents too. So here we've just looked at the distribution of a polygenic score according to socioeconomic background, and you can see that there's higher levels of the PGI in more socially advantaged groups. So if you've got a parent who's got a degree, you're more likely to have a high polygenic score than if they had no educational qualifications. Education is only one of the traits that has changed dramatically over the period captured by CLS's cohorts. There's also traits like smoking and BMI. We're all aware of the obesity epidemic. On screen is the trend in the distribution of BMI among 11-year-olds in the UK since 1991. I just put this on screen to highlight a perhaps less known feature of the obesity epidemic, which is that the underlying population distribution of BMI hasn't changed uniformly. In fact, it's become more variable and more skewed. So there's higher levels of obesity, but very little change in the levels of underweight. Now, on the face of it, this might suggest that there's heterogeneous effects of the obesogenic environment. So in another paper using the cohorts, we investigated whether the association between a polygenic score for BMI and phenotypic BMI had changed over cohorts. And we were also able to use the repeat measurement of BMI in each of the individual cohorts. So we're able to look at associations from birth or young childhood up to age 69 in the oldest cohort. In this study, we're able to also use data from the Millennium Cohort Study. And this is really beneficial because the difference between the Millennium Cohort Study, which is the pink line, and the 1946 cohort, which is the blue line, in the food environments that they grew up with is particularly stark. For instance, the 1946 cohort grew up during post-war rationing. I want to just focus on the results in the top left-hand plot, which show the association between the polygenic score and phenotypic BMI by each cohort and follow-up combination. So for instance, this dot here, if you can see my cursor, I don't know if you can actually, so if you can't, is the age 69 result in the NSHD, the 1946 cohort. And we see that at a given age, the associations are much stronger in the more recent cohort, particularly if we're looking at the Millennium Cohort Study versus the other cohorts. In this paper, we're also able to use quantile regression, and we showed that the cross-cohort differences were particularly stark at the upper centiles of the BMI distribution. So there's stronger differences between these lines at the 90th centile of BMI compared to the 10th centile of BMI. So the differences in obesity are large, but the differences in the levels of underweight aren't large. As I spoke about before, we might be interested in the effect of family environments on children, for instance, the effect of parental education on child education. But we know that children inherit genes as well as environments from their parents, so the association might be confounded. So father's education and child's education is correlated partly because it might have a causal effect, but also because it's confounded by direct genetic transmission of the father's genotype onto the child's genotype. Thankfully, though, if we have genetic data from families, i.e. trios of mothers, fathers and offspring, we can account for genetic transmission using the polygenic scores that we have by simply conditioning upon them. So if we condition on the child's genotype, we can look at indirect genetic effects, or what's sometimes called genetic nurture effects, which should operate only via the parental phenotype. So here we're using genetic data to look at environmental effects, even though we're not actually interested in genetic effects per se. They're just a nuisance factor. In the study that we actually performed, it wasn't education. We looked instead at the associations between familial BMI, so father's BMI, child's BMI, and mother's BMI. And we used the genetic trios data from MCS and used repeated measurement on BMI from children across childhood. So that's from 3 years old to 17 years old. In this setup, we use polygenic scores as instrumental variables in what is using a method called multivariable Mendelian randomization. So the results are on screen now. The green, they're split by whether we were interested in mother's BMI or father's BMI being on the right-hand side. The green dots show the association, the phenotypic association between father's BMI and offspring's BMI by age, and the orange points show the Mendelian randomization results. So if we're just interested in phenotypic correlations, we see strong and consistent associations between father's BMI and offspring BMI. But then if we use the multivariable Mendelian randomization method, these associations are null. So there's no evidence of association once we control for genetic confounding. If we look at mother's BMI, we also see a consistent observational association phenotypically. So a mother with higher BMI is likely to have a child with higher BMI as well. When we use the multivariable MR method, we also see a consistent association. So I just want to finish for completeness talking about another way that you can look at this indirect genetic effects. And this is using a method from Augustine Kong, proposed in 2018. If you have trio data, you can observe which genes a parent passed directly onto their child and which ones they didn't. You can then make a polygenic score of non-transmitted genes to look at the effects that can only have occurred via the parents' phenotypes or from the environment. Now, if you wanted to do this type of analysis, you would need to apply for the raw genetic data, but I think it's a very interesting paper that you should read. Okay, back to you, Tim.
Okay, so acknowledgements to funders, ESRC, who fund the Centre for Longitudinal Studies, and we're hosted by the UCL Institute for Education. And then acknowledgements to Gemma Shirby, Ida Sanchez, and Andrew Wong, who helped out hugely with a lot of the genotyping, did a lot of the QC work, and to the PIs of the studies, including the 1946 study at the bottom. That's it for slides. We'll move on to a Q&A session.